Okay, ladies, let's continue where we left off. Okay. You guys have a good day today so far? Beautiful weather out there. It's cold. Yeah, me neither. So you should get a day off. Last time yesterday I showed you a picture of the number of chromosomes. We looked at why humans have a pair of chromosomes less than chimpanzees, and I showed you the genetic evidence that two chromosomes actually fuse together. And you saw it. I left a folder up there if you ever want to look at it again, where you can actually see where they actually get stuck together, glued together. So that's where we left off, and because we were talking about how do you make things become different from something else, which is basically how do you take a group of organisms and make one, like split them in some way so that they become different from each other. And we said that we need one more thing. We need isolation, right? You need to be able to split them some way. And we talked about you can split them physically. So maybe there's some sort of a barrier. So that's, maybe they're split that way. Maybe it's genetics. So the example that I showed you yesterday with the humans having one less chromosome all paired up, that would be an example of something that's genetically isolating the population. It's nothing physical. And if it's not physical, then it's sympatric speciation, right? So we left off talking about isolation is important because you need to have some barrier to preventing things from breeding. So I was looking up for some of you asking me about, and I try to keep it simple and say, stuff like, if they don't have the same number of chromosomes, they might not be able to pair up. But it's not just the number of chromosomes too. Like I believe donkeys and horses have the same number of chromosomes. I believe. If someone wants to Google it, you can take a look. So it's not just the number of chromosomes. There's other things in there too. Okay, so I'm just saying, keep it simple. I don't want to go into too much detail, but there are other things like mutations that we see in donkeys and we see in horses. And if you look at horses, they're like homozygous for this type of mutation, so they can breed with each other. And donkeys are homozygous for a different type of mutation, so they can breed with each other. What happens if you get something that's got a mutation from each side, a horse and a donkey, even though they have the same number of chromosomes? The fact that they're heterozygous for those two types of mutations makes them incompatible. Okay? So incompatibility is not just the number of chromosomes. It's more complicated than that. But, that's just, I don't want to go into too much detail with that. The idea is you need isolation, okay? And when you give things long enough time, they will start to exhibit big change. And that's what we're talking about now, macroevolution, okay? So these are big changes. And we said that it can happen with a slow accumulation of little changes. And I give you an analogy with changing the dancers. But today what we're going to talk about is how do you make big changes with little changes, okay? Not an accumulation of lots of changes, just maybe one or two little changes. So, for example, things like feathers. What do you think feathers evolved for? What do you think feathers evolved for? Yeah. Why do you think animals evolved feathers?
They were cold.
How many do you think it's for flight?
Okay, but
No, that's what I would guess it was for flight, right? Because you see things with feathers for birds, and typically birds can fly. But what's funny is a lot of dinosaurs. You guys seen Jurassic Park?
I love that.
Yeah, a lot of dinosaurs had feathers, and they weren't flying. So those feathers did not evolve for flying. They evolved for some other purpose. But feathers is an example of macroevolution. It's a big change, okay? You're taking, because feathers are scales. Feathers and scales are the same thing, but they've been dramatically modified. So that's an example of macroevolution. So what we're going to do today, we'll talk about how do you make big change. One, I said it's an accumulation of little changes, but today we're going to talk about a different type of science, okay? We're going to talk about something called Evo-Devo.
What?
Evo-Devo. Which stands for evolutionary development. We already looked at this. We talked about this as an example of something that its development has changed. And that's made a really dramatic impact because if you remember in this scenario, the gills are kept, they're retained.
But that's a real thing.
Yeah.
Not that thing again?
Yeah. We talked about T-Rexes having the little forelimbs. That's an example of changing development to make a big change. We talked about how in the inner fish video, we talked about how our human brain development has been slowed down, how our facial development has been slowed down. Changes in development produce big changes, okay? Those are big changes. A mutation that allows you to digest milk, that's not a big change. You can't really see that. It's a very small change. It's important, but that's not a big change. So let's talk about big change. Talk about Evo-Devo. What have you noticed? Something unusual about this dolphin.
Why does it have those thingies?
It actually has an extra pair of fins back here. You see this? That's an extra pair of fins. Now, dolphins and whales have the front flippers, but they don't have these. What happened here? What's going on? Okay? At the end of it, let's see if you can figure out what happened. So I'm not going to explain it right now. What we're going to do is we're going to talk about genetics again, but from a different point of view. Because we focused on mutations that affect a gene that changes the protein in some way. So, for example, what was the problem with sickle cell anemia? What protein was modified? Very good. Hemoglobin. Okay? In that case, the allele gives you a protein that is different. What was the problem with cystic fibrosis? Remember what the protein that was modified? It was a protein that allows the transport of what? No, it's actually chloride, which then affects water. But again, it's the protein that's deformed in some way. Okay? But lactose tolerance was different. The protein is not what's being modified. What's being modified is the regulation. So how many of you know what the term regulation means? Means, what's another synonym for regulation? Control. Control. Another word for regulation is control. So you have to understand that a lot of your genes in your body are controlled. They're switched on at certain times and then they're switched off. With lactose tolerance, the gene is left on. If you're lactose intolerant, it's on initially, then at around 4 or 5 years of age, it's switched off because we're all mammals, and we talked about mammals start off drinking what? Milk. But then they stop doing that. It's switched off. So I need you to think of a gene as something that can be switched on and off. Okay? You understand that? All right. So a gene can be switched on and off. And we need to also think of two types of genes. So now remember I give you the analogy about the dance and the dancers and the choreographer? Okay. There are your regular genes that do things like build hemoglobin, allow you to taste for pt2 or sodium benzoate, allow you to see color, allow you to move chloride. Those are regular genes, okay? But then there are the genes that control the other genes. Okay? They are the genes that control the other genes. And they're called the regulatory genes, or you can call them toolkit genes because they act like a toolkit. They are involved in building stuff. So these genes are like the choreographer. They control the other ones. So far, so good? Okay. Because what we're going to focus on are not just regular genes. We're going to focus on toolkit genes. And these are the genes that control other genes. I'm going to show you some examples. Okay? One thing that scientists have figured out over the last few decades is a way to make a gene visible. So they have a trick where they can do stuff where the gene actually shows up visibly, and you can see it here. So you see in this picture here, you see these banding patterns. Okay? These banding patterns are areas of where a gene called GDF5 is working. Okay? You see this? That's all highlighted. This is a gene called scleraxis, and it's working in this area. You see this that's highlighted? It's a purple color. This is a gene called BMP4, and it's working in between the fingers. And here, you see these little purple dots? That's a gene called patched. So the advantage of being able to visualize a gene, you can see where it's working, where and when it's working. I'm going to ask you a question. What do you think GDF5 does? I want you to look at where it's working. Right. It's involved in forming the joints in the digits, fingers. Okay? This scleraxis is involved in building tendons. These are the things that attach to muscle and make them move. BMP4 is working between the digits or between the fingers. Now, what do you see between my fingers? Nothing. Guess what that gene does? We all start off with webbed hands, and that gene actually gets rid of the webbing. Okay? And then patched. This is a chicken embryo. Guess what patched is involved in? Building feathers. These are toolkit genes because what they're doing is they're building the body. Okay? They're building the body, and to build something, well, you need tools. You need tools. So far, so good? Any questions so far? So I'm going to show you some examples of where toolkit proteins have been changed in some way to give you a really big change. Okay? All right? So let's start with a picture. Now maybe you know what this is. It's a snake. What's the other organism?
A rabbit.
It is. Hold on. Let me get the rabbit here. Here's a rabbit. This is that. Now, you'll notice the snake's anatomy is different, but there's a lot of similarity. If you look at the snake, you'll notice that here's the head, and then here's the vertebral column, and then you pretty much see ribs starting from right behind the head. You see the ribs? And they go all the way down the body. But if you look at the rabbit, you'll notice there's a head, and there's a neck. And where the neck ends, the shoulders begin. And then where the shoulders begin, you see rib cage. So you'll notice that the ribs don't start really until the neck ends. See the difference there? I mean, there are other differences like the limbs. We'll talk about that in a second. So you'll notice that you can see in a rabbit, you can see the neck, and the neck does not have any vertebrae sticking out of it. So there's no ribs sticking out of your neck, right? But in a snake, what do you start to see? You start to see those ribs right after the head. What's going on here? So there's a gene. It's called HOXC6, and you can see it right over here. Okay, so here's HOXC6 right here. Now, this is a toolkit gene. So what it does is it's like the choreographer. It instructs other genes, and it says to the other genes, this is where you build the basically the end of the neck. Okay? This is where the neck ends. So it's like a choreographer. It tells other genes what to do. Now, in a mouse and in all mammals, here's the head. Okay? Here's basically where the shoulders begin. And you have 1, 2, 3, 4, 5, 6, 7. 7 what are called cervical vertebrae. That means if you looked at your neck, so if you looked at a rabbit, there would be 7 cervical vertebrae. Okay? 7 of these things. Okay? And then you can see the ribs after the neck. So HOXC6 says this is where the neck ends. And where the neck ends is where the shoulders begin. And where the shoulders begin is where your front limbs begin. Okay? Now, what's the difference between a chicken and a mouse? Chickens have much longer necks than a mouse. And geese have even longer necks. Now, what is it? Is it an accumulation of little changes? Turns out in this case, it's not. It's just one change. It's basically that gene, HOXC6, does not fire. So you got to envision this is an embryo. This whole thing is a developing embryo. Okay? Where these genes are working, you got to think of time and location. It's like, think about you, right? Right now, you're at a specific moment in time, and you're at a specific location on this planet. Every cell in your body has the same genes. But when and where they're working is not the same. So what's going on is that when this embryo is developing, HOXC6, in this case, turns on at a particular moment in time and in a particular spot in the embryo. But if it mutates in some way, it could fire in a different location. So, why does a chicken have a longer neck? Because HOXC6 is working further back in the embryo. It's working here, in the 19th vertebrae, not the 12th. Or if you think of these as units, it's working further back. Why does a goose have an even longer neck? Because it's working even further back. So the difference between humans, chickens, because humans and mouse are the same, and geese, is just simply this gene is firing in different spots. Now, look at the snake. Where does it fire? Pan, you have a question? That's okay. Fire means, yeah, maybe I should say fire. Fire means it's working. Yeah. Yeah. Right. So when it switches on, it says build neck here. Or I should say, when it switches on, it says neck ends here. Okay? So when it switches on, when it fires, it says neck ends here. So for a mouse, it says neck ends here. For a chicken, it says neck ends here. For a goose, it says neck ends here. What's interesting is for a snake, it says neck ends here. Now, is that further back or further forward? Further forward. In fact, it's so far forward, it basically says end the neck where the head ends. Now we have a problem. There's no neck. Exactly. And here's an interesting thing. I want you to understand, one of the things that you, like if you were, how many of you guys have been involved in performances, right? You guys have been involved in assemblies, and one way you know when it's your turn is to know who's going before you. Like, for example, if I'm the 7th act, I don't need to know what time I'm on. I just need to know who the sixth act is. And if the sixth act is on deck and they're performing, I know I'm next. Well, in this case, I want you to think of it this way. When the body's being built, you form the head, form the neck. Where the neck ends, you build the shoulders and the front limbs. Problem is, when there's no neck, how do you know where to build the front limbs? That cue was lost. So losing the neck here actually caused the loss of the forelimbs. They did. A long time ago, at some point, there was a mutation where this gene was expressing itself further back. So the ancestor would have had a neck, but got expressed further forward. Lost the neck, lost the forelimbs. Now, was that mutation an advantage? Yeah, it was, because otherwise the snakes wouldn't be around. Okay? So you can see that by having no neck, then basically, look, we talked about this. Where the neck ends, the ribs begin. That's why the ribs start so far forward in a snake. Okay? It's one tiny little change, but it's in the change of a gene that acts as a choreographer. Okay? So that's explained in this slide. That's the Hawk C6 toolkit protein. Show you another one. This other one is for wing formation in fruit flies. Okay? Now, I don't know if you know this, but fruit flies have two pairs of wings. They have a front wing, which is really big, and then they have a hind wing, which is smaller. Okay? Now, show you a picture of a fruit fly. This is a normal fruit fly, so you can see the big forewings, and then see these little structures right here? They're really tiny. Those are the hind wings. So they're very small. To me, it reminds me of a plane. You know, a plane has the big front wings and then the little hind wings? That's what it kind of reminds me of, okay? This on the right is a mutation. Okay? It's a mutation. What do you notice? Two pairs of wings. Now that's a mutant. If you catch a fruit fly in the wild, you won't find that. Because that thing has very low fitness. Chances are it will be caught by something and it will be eaten. The problem is they're not having this setup. They don't maneuver very well. And if you're a little fruit fly, you want to maneuver really well, okay? Because you need to get away from things. Same is true for a housefly. Now, you notice the colors here? You see different colors, right? You see like blues, magentas, reds. Again, those are genes that are working. Okay? Those are genes that are working. So you'll notice that this is the front wing. This is the hind wing. Does it look different or the same? To look at the color patterns. Because it's the same genes that are working. So the same genes building the front wings, same ones building the hind wings. But there is a gene that behaves differently. This gene, you'll notice, this is the front wing. Okay? This is the hind wing. This is the gene, it's highlighted yellow. You'll notice that the expression pattern here, where it's working, is it the same as where it's working here? It looks different, right? This gene is called UBX. Now, other than size, what else do you notice about the difference between the front wing and the back wing? Other than size, what's missing? Right, these lines. You see the lines here? Okay, notice how it's missing here? These lines give the wing strength. Because what do these wings, these are the wings that generate all the power for flight. So these wings have to be really strong. These wings, they're more for stability, so they're not generating lift. They're just basically steering. And notice the lines are missing. So the difference between this wing and this wing is a simple mutation. Little change. Where you can see that UBX is working in the front wing it's working on the outside a little bit with very little inside. But in the hind wing, it's working pretty much inside the wing and very little on the outside. That little change, okay, that little change produces the hind wing. The original version, if you look at the earliest flying insects, like things like dragonflies, how many sets of wings do dragonflies have? They have 2. So, at some point in the evolution of certain insects, that toolkit gene underwent a small mutation where the hind wing was changed because that gene, you can see, starts to work in a slightly different location. Again, change the choreographer, you make a big change. Okay? It's just due to one little gene, but that gene has a big effect. Okay, so you can see it here. So here's the forewing, okay, and then here's the hind wing. Right, these are just the expression patterns of the gene. So you can see it's the same gene, okay, it's the same gene, but it's just doing something different because it's working in a slightly different location. Okay, let me show you another example. And that's explained over here, okay? Stickleback fish. Okay. Now, this is the what's called a heavily armored version. Think of it like your tank. Okay? This is the lightly armored version. Think of it like a car. Okay? You'll notice that this one has these big spikes, and this one has these tiny little spikes. Okay? Turns out that the difference between these two comes down to one gene. It's called pt2. Now, the thing about toolkit genes is that because they're involved in building the body, they typically have roles in many locations. So, let me explain this diagram, what this diagram means. So this is the actual protein. So, remember what a gene gives you? It gives you a protein, okay? Like hemoglobin. But a toolkit gene gives you a protein that controls other genes. So the protein that it makes is, you'll learn about this in grade 12. It's called a transcription factor. And it controls other genes, okay? So it's not the protein that's being changed, it's the switch. The switch is what controls when and where that gene works. This tells you where the gene works. It's telling you that it's working in different locations, okay? You'll notice the difference, but it's more than this. You'll see that the difference between this fish, the heavily armored, and this one is that a mutation has disabled it in this part of the fish, the pelvic fin, which is here, in this band. Okay, so here's your pelvic fin region, right over here. So, it's working in the thymus, which would be basically in this area. It's working in the lateral sensory organs, which runs along the length of the fish. But it's not working in this region. So, the difference between the heavily armored version and the lightly armored version is one gene that gets disabled when this pelvic fin zone is being developed. That's the only difference. One little change. But it produces a big effect. Here's the question. Natural selection predicts, or Darwinian evolution predicts, that if a mutation produces something that's favorable, you will see more of it, right? Which is the better? So which is better? Yeah, see, I can't get you guys anymore. So, it depends where you are. Depends who your predator is. If you're out in the open, so what they did was they looked at what are called the oceanic form out in the open water, and they looked at what's called the river form, or the ones that we find in streams. smaller bodies of water, okay? In the open water, the main predator of the stickleback are big fish. But in streams, the main predator of the stickleback are dragonfly larvae. Now, dragonfly larvae, I don't know if you know this, the babies start off in the water, and did I show you this? I think I did. Yep. A few inches. They're tiny. I've never seen you. Okay, after this video, I'll show you the dragonfly larvae. They're nasty. They're very nasty insects. Okay, and they hunt frogs. They hunt fish. What's better, to be heavily armored or to be lightly armored? According to this diagram, if you're living out in the open water, your main predator is the fish. It's better to be heavily armored because it might prevent the big fish from swallowing you. Because the big spikes will hinder the big fish from swallowing you. But if you're living in a stream and you're got these big spikes, according to this picture, that could be a problem. Because guess what the dragonfly larva can grab? The big spikes. In that case, being a tank is not a good idea. So again, you guys are knowing this right now. What's an advantage? It often depends on the environment that the organism lives in. Okay? So what you find is the oceanic form, you're going to see they'll have more spines, or the spines will be bigger, but in the freshwater form, in the streams and rivers, it'll be the opposite. Okay? We're almost done. Any questions? No? Okay. So we talked about snakes. We talked about how Hawk C6 is the reason why they lost their neck. The hind limbs, however, is a different scenario. Okay? The hind limbs includes a bunch of different mutations. One that may be familiar to you. Do you recognize Sonic Hedgehog? Where did you see that? In the video. Do you remember what it does? It's involved in building what? Fingers, okay? So the loss of the hind limbs has nothing to do with Hawk C6. It's a different scenario. One of the genes that is behaving differently is Sonic Hedgehog, which we saw in the inner fish, okay? Do birds have teeth? Did they? They had to, because what are birds? You know what they descended from? Dinosaurs. And what do dinosaurs have? Big scary teeth. So the thing about evolution, it makes certain predictions. You can predict what a transitional form from water to land would look like. And we saw that with, what was the organism? Tiktaalik, right? You should also be able to predict that birds don't have teeth, but we know they came from dinosaurs, so they must have had teeth at some point. So there should be proof in their genetics that they can make teeth, and here it is. So this is a tooth from a hen. Now, chickens don't have teeth. For some reason, it was natural selection favored no teeth in birds. I don't know why. No idea. Maybe the dental bills are too expensive. They don't. They swallow it. And then they use like stones to break up the food. But here is a bird that was coaxed into making teeth. And what they did was a very simple experiment. They took tissue from a mouse, from the mouth of a mouse, developing mouse. And they transplanted the tissue, and this tissue, by the way, is from the part of the mouth that produces the teeth. Okay? So they transplanted the tissue into a developing chicken, and they put it in the mouth of the chicken. Okay? And what happened is the chicken grew up with teeth. Now, birds haven't had teeth for at least 100 million years. This kind of reminds me of the experiment you saw in the inner fish, when they did a bit of a transplant. and the bird ended up having extra digits. What this is saying is that even though birds don't make teeth, they still have the ability to make teeth. But the programming has been switched off. Okay? It's there. It's still there. The programming to make teeth is there. It's been switched off. Okay? So an example where a toolkit gene has been disabled. Now the gene that was disabled is BMP4. Okay? In birds, where the teeth are forming, it's turned off. I showed you before that BMP4 is also involved in building what? What was it also involved in? Building the... Yeah, right here. Involved in building the hand, right? Just a reminder that toolkit genes often work in many locations. Okay? Often work in many locations. So basically the idea here is that maybe they're switched off in one part of the embryo, but they could still be working somewhere else. Okay? And that's what you're seeing here. Is that kind of cool? So I'll end up with this. You guys have seen Jurassic Park, right?
No.
You ever seen the original one?
No. We should watch it.
No?
Let's watch it.
So the original one, they have this defined dinosaur DNA, and they fill it in with frogs DNA. I think it's frogs DNA. If you had to build a dinosaur, what organism would you fill it? What organism would you use? I would use a bird. In fact, there was a book written by a guy named Jack Horner. It's called How to Build a Dinosaur. And he argues that because birds are evolved from dinosaurs, you can almost, if you know the genetics, like we know that BMP4 is switched off in the mouth and you don't get the teeth. Guess what happens if you switch it back on in the mouth? You get birds with teeth. If you can know all of the changes that occurred that gave you birds, what would happen if you reversed it? You will get something like a dinosaur back. Isn't that kind of cool? I'm not condoning that we do this.
Can we do it?
But it is, I think it's a little bit beyond my ability. Anyways, ladies, that's it for macroevolution, okay? The end.